Medical nanotechnology, revolutionizing targeted drug delivery. Introduction. The field of medical nanotechnology has emerged as a groundbreaking approach towards improving drug delivery systems in treating various diseases, including cancer. This script aims to provide a detailed overview of key aspects in medical nanotechnology, including the tumor microenvironment, passive and active targeting strategies, pharmacokinetics, drug release mechanisms, multifunctional nanocarriers, stability and biocompatibility, overcoming biological barriers, regulatory aspects, preclinical models, clinical trials, and future perspectives. By understanding the intricacies of these topics, medical students will gain valuable insights into the potential of nanotechnology in revolutionizing targeted drug delivery. The tumor microenvironment plays a critical role in cancer progression and response to therapy. It is composed of various components, including cancer cells, stromal cells, extracellular matrix, and blood vessels. Nanoparticles can be designed to exploit the unique characteristics of the tumor microenvironment, such as enhanced permeability and retention effect, EPR, to achieve selective accumulation in tumors. Additionally, the incorporation of targeting ligands on nanoparticle surfaces enables specific interaction with tumor cells, further enhancing drug delivery efficiency. Passive targeting refers to the accumulation of nanoparticles in tumors through the EPR effect. The leaky vasculature of tumors allows nanoparticles to extravasate and accumulate within the tumor parenchyma. Furthermore, the impaired lymphatic drainage in tumors contributes to the prolonged retention of nanocarriers in the tumor microenvironment. This passive targeting strategy offers improved drug localization, reduced systemic toxicity, and increased therapeutic efficacy. Active targeting involves the use of ligands, such as antibodies or peptides, to specifically bind to tumor-specific receptors, achieving enhanced selectivity and accumulation of nanoparticles within tumor tissues. The ligands can be conjugated to the surface of nanoparticles, facilitating receptor-mediated endocytosis and intracellular drug release. Active targeting strategies enable improved tumor penetration, bypassing biological barriers, and maximizing therapeutic outcomes. Understanding the pharmacokinetics of nanocarriers is crucial for optimizing drug delivery efficiency. Factors such as particle size, surface charge, and surface modification influence their in vivo behavior, including circulation time, biodistribution, and clearance. Nanoparticles with appropriate physicochemical properties can enhance drug stability, extend systemic circulation, and increase drug accumulation at the target site, ultimately improving therapeutic outcomes. Nanocarriers play a crucial role in achieving efficient drug release to solid tumors. It is important to consider various drug release mechanisms in nanotechnology. These mechanisms involve diffusion, degradation, and stimulus-responsive release. While diffusion involves the gradual release of drugs from nanocarriers, degradation refers to the breakdown of carrier materials to facilitate drug release. Stimulus-responsive systems, on the other hand, allow for targeted drug release by responding to external factors like temperature, pH, or enzymes. Such controlled drug release mechanisms enhance treatment outcomes in solid tumor therapy. Multifunctional nanocarriers are advanced nanosystems that combine several essential features into a single platform. They are specifically engineered to simultaneously target, image, and treat solid tumors. By incorporating therapeutic agents, imaging agents, and targeting ligands, these nanocarriers enable personalized medicine by allowing real-time monitoring and precise drug delivery. Their versatility is key in overcoming the challenges of drug resistance and optimizing the effectiveness of therapies. In the field of medical nanotechnology, it is essential to establish the stability and biocompatibility of nanocarriers for their successful application in treating solid tumors. Nanoparticles must possess long-term stability, meaning they should retain their physical and chemical properties even when stored and circulating within the body. Furthermore, it is crucial to ensure that these nanocarriers are biocompatible, meaning they do not cause any harmful effects on healthy tissues. Rigorous evaluation of the stability and biocompatibility of nanocarriers is necessary to guarantee the safety and effectiveness of these treatments in patients. The successful delivery of nanoparticles to the target site faces challenges posed by biological barriers, such as the reticuloendothelial system, renal clearance, and cellular uptake mechanisms. Strategies to overcome these barriers include surface engineering to evade immune recognition, size optimization to avoid renal clearance, and formulation approaches to enhance cellular uptake. Overcoming these barriers can significantly enhance the therapeutic efficacy of nanomedicine. As with any medical innovation, regulatory approval is a crucial step in the translation of nanomedicine into clinical practice. Regulatory agencies evaluate the safety, efficacy, and quality of nanomedicine products through rigorous preclinical and clinical testing. Compliance with regulatory guidelines and standards is essential to ensure patient safety and gain market access. Understanding the regulatory landscape is crucial for the successful development and commercialization of nanomedicine. Preclinical models are important tools used in the evaluation of nanocarriers' effectiveness and safety prior to human trials. Through the use of cell-based assays and animal models, scientists can gather valuable data regarding the kinetics of drug delivery, biodistribution patterns, and potential toxicities. These preclinical evaluations play a crucial role in guiding the design and optimization of nanocarriers, ultimately facilitating their successful translation into clinical trials. A rigorous and thorough preclinical assessment is therefore imperative in ensuring the advancement of nanoscale drug delivery systems in the fight against solid tumors. Clinical trials play a pivotal role in assessing the safety, tolerability, and efficacy of nanomedicine in human patients. Phase 1, 2, and 3 trials are conducted to evaluate the pharmacokinetics, therapeutic response, and long-term effects of nanocarriers. Clinical trials involve rigorous evaluation of nanomedicine's bioactivity, toxicity, and patient outcomes, leading to regulatory approval and subsequent commercialization. Looking ahead, the future of medical nanotechnology holds immense promise and potential. Advancements in nanomaterials, fabrication techniques, and personalized medicine will further enhance the targeted delivery of therapeutics. Novel imaging modalities, such as nanoparticle-based imaging agents, can revolutionize early disease detection and monitoring. Moreover, the integration of nanotechnology with gene therapy and immunotherapy opens new avenues for advanced treatment modalities. In conclusion, medical nanotechnology offers unparalleled opportunities for targeted drug delivery. The intricate interplay between nanocarrier design, tumor microenvironment, targeting strategies, pharmacokinetics, and regulatory aspects determines the success of nanomedicine. By harnessing the potential of nanotechnology, we can revolutionize the treatment of various diseases, including cancer, ultimately improving patient outcomes and quality of life. As medical students, understanding these key aspects will prepare you to embrace the future of medicine and contribute to the advancement of targeted drug delivery.